So for fertilization to occur, it is actually conducive here because both the Antheridia and the Archegonia are nearby, since it is monoecious, and also the fact that the Antheridium develops just behind the archegonial receptacle. So the close proximity of the male and female receptacles ensures fertilization. But still they have seen that it occasionally it fails to occur. Okay, so they have seen that the motile sperm undergoes a change, okay, from the time it leaves the antheridium until it is ready to fuse with the egg. So uh, sometimes when the first sperm nucleus penetrates the egg cytoplasm, it lies close to the egg nucleus, and at this time the egg nucleus has about twice the diameter of the sperm nucleus. Okay, and apparently the fusion passes into the resting stage before division of the egg takes place. So this is the mature egg and how the embryo is formed. So this shows the egg with the male and the female nuclei. Okay, and this is the oil drop. Okay, and first division is transverse. Okay, and you have the mature egg and the early embryo are elongated. Okay, they are elongated, slightly curved. Okay, bilaterally symmetrical bodies. So fertilization takes place in October. Development of embryo begins at once, and the sporogenous tissue becomes differentiated by winter. Okay, sporophyte matures in May and June. So there's a gap of some 3 to 4 months before between the fertilization and the maturation of the sporophyte. So this is the steps how the sporophyte gets developed. So early embryo, the first division is transverse. Okay, it forms the hypobasal cell and the epibasal cell. The upper cell is called the epibasal cell. The lower cell is called the hypobasal cell. So it divides in such a way that it forms 4 cells. Okay, okay, vertical wall formation occurs. Then you have, after the transverse wall formation, you can see vertical wall formation which occurs. It is oblique wall. Okay, conal இருக்கு பாருங்க. It is oblique wall. Vertical wall is oblique. Okay, commonly in the basal cell you can see the vertical wall development. Okay, so this is to cut the foot and the stalk portion. So this portion forms the foot. Okay, to the vertical division mainly occurs so that the foot portion is demarcated. Okay, so the foot and the stalk are derived from the hypobasal cell, the lower cell or the hypobasal cell, and the epibasal cell giving rise to the capsule. This is the epibasal cell giving rise to the capsule. Okay, basal cell of the row of 4 varies. Now this forms the basal cell, varies in its contribution to the tissue of the foot. Okay, sometimes you have a stalk, sometimes you don't have a distinct stalk. You have very short stalk. So this set of diagrams gives you an idea how the embryo is developed. So immediately after fertilization, the embryo starts to develop. It first starts as a 2-cell stage, hypobasal and epibasal. The hypobasal cell undergoes a vertical division to differentiate the stalk and the foot and the seta, and the epibasal cell undergoes divisions to form the capsule. So these are the few developing stages of the embryo. So as the sporogonium develops inside the capsule, possesses a spherical capsule, seta and foot. Capsule has a single jacket layer. It has a single jacket layer and dehisces irregularly at the apex. Spores are spherical, brown and reticulate. You have elaters are also seen, not numerous. Okay, so the carpocephalum is hemispheric. That is why it is called hemispherica. Okay, proximal is half lobed. Okay, each lobe with 2 slit and one containing 1 sporophyte. This is the, you can see the carpocephalum, hemispherical. Okay, and it opens with a slit. Sporophyte seta is not elongating. I told you some in Reboulia you have very short seta, and capsule is spherical, irregularly circumscissile, and the distal portion is disintegrating. Capsule wall without any thickenings. Capsule wall does not have any thickenings. Okay, and spores are spherical in structure, and you have 2 to 4 elaters. So the life cycle is similar to what you see in Markantia. It has a distinct sporophytic stage and a gametophytic stage. Okay, so the gametophytic thallus develops the male and the female gametophyte respectively. Female gametophyte gives rise to archegonia, male gametophyte gives rise to antheridia. Antheridia develops the sperms and archegonia develops the egg. So when fertilization takes place in the presence of water with the help of the mucilage which are secreted by both archegonia and the antheridia, the egg is it fertilizes the egg, and the sperm fertilizes the egg, the venter, and development of sporophyte begins. The development of embryo begins immediately after fertilization, but the sporophyte develops only after 4 months. So the development of the embryo to a sporophyte takes around 4 months time. And the spores, you can see the spore capsule on the underside of the archegoniophore, as I had told you in the shown you in the picture. And the spores are released, each spore accordingly the male genes or the female genes, they develop into the male gametophyte or the female gametophyte. Mostly they are bisexual spores, I mean monoecious conditions, so the same thallus will bear the male and the female sex organs. So to summarize, Reboulia comprises of a single polymorphic species, Hemisperica, belonging to operculate division of the subfamily. The thallus bears smooth and pegged rhizoids. So that's smooth and tuberculate rhizoids, and 2 ranked ventral scales without appendages. The body is differentiated into a dorsal and a ventral region. And air chambers are abundantly formed, developed by centripetal splittings. Secondary partitions separate the primary air chambers. No chlorophyllous filaments are formed. Barrel-shaped air pores are developed on the female receptacle. I had shown you the air chamber of the female receptacle, which are barrel-shaped pores. And those of the thallus and the male receptacle are made of a single layer of concentric cells. That's what I had told you here. I had shown you the thallus as well as the antheridial receptacle. Male receptacle has a very simple pore. So further summarize, it is monoecious or autoecious you call it. Antheridial receptacle is sessile. Several may be produced, but the formation of archegonial receptacles terminates the apical growth of the thallus. So once the archegonial receptacle is formed, thallus growth is stopped. Thallus does not grow further. So it represents a definite branch. It stops growing. So growth of limited, the growth becomes limited. Growth point becomes limited. The thallus stops growing at that point. Antheridia develops like those of other marcantiales. Antheridia are usually, they are developed in the deep-seated tissue of the dorsal surface of the thallus. So you have in the development of archegonium, 3 vertical walls follow the appearance of a transverse wall. As I had told you during the development of archegonium, how the horizontal divisions and the vertical divisions form a long neck region, okay, and swollen venter portion. So several growing points are organized in the female receptacle from segments of the apical cell. And so based on how many growing points are there, you have so many lobes found in the female receptacle. So they have seen 4 to 7 lobes in different subspecies. So each lobe corresponds to a growing point on the thallus. And one orchigonium is formed from the apical cell. So that finishes the Reboulia hemisphaerica life cycle.